First of all, thank you, Ilya, thank you, organizers, for this opportunity to finally speak at this very nice conference. My name is Maxim Skulachev. I'm PhD in molecular biology and Chief Scientific Officer of Mitochondria-Targeted, the mitochondrial company. And we are focused on this beautiful organelle, and thank you very much, Shai, for introduction of mitochondria and how important and how cool it is. And now it's generally accepted that mitochondria play some important, and according to many scientists, one of the most important roles in the process of aging. And my scientific supervisor and my father, Vladimir Skulachev, who passed 2 years ago, was absolutely certain that mitochondria also play a role in so-called the program of aging. Well, I will not go into the discussion whether aging programmed or not. We don't want to do it. But anyway, mitochondria are important for aging, and that's our target. How do we target it? By the nature of mitochondria. When they burn substrate, the first thing they do, they charge the membrane with enormous electric charge. Actually, it is the only negatively charged organelle in our cells. And it was proved in 1969 in the laboratory of my father back in Moscow State University by adding very special substances to mitochondria, penetrating ions. These are charged ions, they're positive, but contrary to all other ions present in our body and in nature, they can penetrate through any membrane. So if they see anything negatively charged, they will accumulate there. And there is only one place in our cell, negatively charged is mitochondria. So actually the idea is pretty simple. You take such penetrating ion. Actually, some American scientists call it Skulachev ion, but usually it's called lipophilic or penetrating ion. And you attach something you want to target to mitochondria to it. What do we want to target? Antioxidants. Because one of the major dysfunctions of mitochondria, production of reactive oxygen species. And we want to fight with this problem. So we designed this molecule, it's called SKQ1. Actually, we designed many molecules of that type, and more are coming. We're working, so we are trying to optimize the design of our molecules. And they really target mitochondria. So some of our SKQs, they're fluorescent. You can look in the microscope. You can stain mitochondria, for example, with YFP, and you have perfect match. The drug really goes into mitochondria and only into mitochondria. One more feature of these molecules, they're rechargeable. Usually when antioxidant meets ROS, meets radical, the antioxidant is being oxidized. It reduces and neutralizes the thread, the ROS, but then it's wasted because it lacks electrons. Our molecules designed that way that they can actually get, after oxidation, extra electrons from electron transport chain of mitochondria. And we know exactly the place in complex 3 where this process goes. So it is antioxidant which can be restored in mitochondria and used again and again. That rises tremendously the efficiency. The first target for our drug in the cell is cardiolipin. It's unsaturated lipid present only in mitochondria, which being burned by oxidative stress first in our cells. And we demonstrate experimentally how cardiolipin is very nicely protected by SKQ1 in model experiments with oxidative stress. You can even see it on cellular level. If our cells feel good, mitochondria actually they form long threads. It's kind of electric wires which sometimes connect one part of the cell with another with electricity. But if you induce oxidative stress, these threads they disassembled into little balls, little grains, and then starts some quality control of mitochondria. Some of these grains just disappear, being eliminated. So mitochondria, they actually live in this equilibrium of thread-grain transition. And if you add SKQ1, mitochondria are very happy, they form these threads. We had a lot of experiments with this compound on animals, on different models, and it really slows down aging of mice, which is not that interesting. Again, I agree with Shai completely. But really, you can delay aging of mice, well, not completely, they are not living forever. There are certain problems, but it works as anti-aging drug as predicted. And at this point, we faced exactly the same problem our colleagues are facing with Cloto technology and others. If you have something which anti-aging potential, how can you administer to humans? This is unnatural compound created by just scientists. It's chemistry, small molecule, but still new chemistry. It cannot be food supplement or something. So it can only come into the organism of a human as a drug. But for drug, you need indication. And aging is not an indication, it's not a disease. So we decided to find some disease, age-related, and try to cure it with the compound as a first step and as a break through the regulatory restrictions to the clinics. Actually, there is one drug, but it's maybe not that interesting, which is already approved in some country and on the phase 3 on clinical trials in the United States. It's eye drops with this compound against age-related eye diseases. But it's local administration, it is not that cool. What we need systemic administration, we need a pill with SKQ to see if it really slows down aging of humans. What indication? Actually, the mechanism, and I mentioned cardiolipin, dictates some indication to apply to SKQ1. There is a relatively novel mechanism recently discovered a little bit more than 10 years ago called ferroptosis, the mechanism of cell death. It turned out that in certain conditions related to overloading of cells with iron, cells start a so-called Fenton reaction. It is very aggressive production of reactive oxygen species going on around mitochondria, oxidizing lipids, and many diseases, the pathological mechanism is linked to this ferroptosis. And this is exactly the indication for our SKQ theoretically. And what is also suspicious, there are more and more papers coming out linking ferroptosis and aging. Seems like it really increases with age, which is not surprising for me. But there is a special disease related exactly with ferroptosis. It's called Friedreich ataxia. It's rare neurodegenerative disease linked to some mutation which causes disbalance of iron accumulation in the cell. And the cells of these patients, especially neurons, are extremely sensitive to oxidative damage, and it causes huge problems to these unfortunate people with these mutations. You can take fibroblasts from these patients, and we show that they are much more sensitive to ferroptosis inducers. You can induce it by certain stress in the cell, actually interfering with glutathione system. And these cells start to die. And fibroblasts from these Friedreich ataxia patients, they are much more sensitive to such ferroptosis triggering. I'm very much proud that this work was done in our Israeli branch of our company. Generally, the company is British, but we have our team here in Israel, and we showed this sensitivity. Also we showed that SKQ1 is extremely efficient in protecting these cells from ferroptosis. You can add as small as 10 nanomolar concentration of our compound and still rescue some cells. Actually, you can achieve the same results with the classic antioxidant. It's oxidation-related death of cells. Idebenone, one of the most potent antioxidants known, but you need to pour 100 times more. This is the power of mitochondrial targeting. You need much less compound to achieve your result. So nice results, and we have positive results with some other indications, neuroinflammation, inflammatory diseases, but it's completely useless because to switch to clinical trials, you need to raise investments, a lot of money. And after the COVID, ones of you who are connected to biotech business will know that it's almost impossible to find investments for new drugs, except one area. Let me tell you, spend 3 minutes about mitochondrial uncoupling. There is another mechanism going on in mitochondria, which is called uncoupling, oxidative phosphorylation, and respiration. What does it mean? Mitochondria, they do two things mainly. They oxidize substrate, charge the membrane, and then use this electric charge to produce ATP. These two processes, charging and ATP production, are coupled. But there are compounds, there are ways to break this link and just dissipate charge into actually heat. It's called uncoupling. I'm extremely proud that this process was actually predicted and described by my father. Actually, for ones of you who know Russian, there is a proof. It's a paper from 1960 in Biochemia journal, a paper by two young PhD students, Vladimir Skulachev and same year of university, his friend Sergei Maslov. They were 25 years old at that time. Speculating mainly, that sometimes maybe the respiration could not produce ATP, but produce heat. And that's the way to heat, just to warm your body if needed. 30 years later, Vladimir Skulachev again published a paper describing very elegant mechanism of just background uncoupling of the cells or organism using free fatty acid cycling through mitochondrial membrane. Free fatty acids, they can actually transport proton and discharge mitochondria. That's why it's, and it is exactly uncoupling and warming up. Then 20 years later, 2010, this is our PNAS paper, we discovered that our SKQ compound can actually transport free fatty acids anion through mitochondrial membrane and stimulate this natural uncoupling. I'm extremely proud of this paper. This is my father, this is me, this is my eldest brother, Fedor Severin. We have different fathers, and this is my mother, Inna Severina. So it's completely family business on mitochondrial uncoupling. And I will not go into the detailed mechanism. Believe me, it does uncouple. So what? Where it can be applied in medicine? Obesity, of course. Because when mitochondria are discharged, uncoupled, they are looking for most efficient substrate to restore its charge. What is this substrate? Fat. All the uncouplers, they are fat burners. So we did these experiments in England in Signature Discovery Company, which are specialized on very obese mice. They weigh about 50 grams, contrary to 20 normal grams. So it's actually balls of fat. And we treated them with SKQ, and they lost about 15% of their weight in roughly 1 week of treatment. And what is very important, contrary to GLP-1 analogs, this weight loss was precisely due to loss of fat. None of bone or muscle mass was reduced in these animals. We recently repeated this experiment in larger animals and non-rodents, rabbits. And again, it is very obese rabbits. And we managed to, with longer treatment, but this is oral treatment, by the way, dose dependently, we managed to decrease their weight by 27%. It's one of the world records, in, even better than GLP-1 agonists in the rabbits. And this indication attracted interest of investors because of semaglutide, Ozempic, thanks for them for opening this huge market. These kind of drugs are interesting for the market at the moment. We tried combination with GLP-1 agonist, and SKQ is actually, it works very nicely in combination. I hope very much that it will help to preserve weight of the patients even with GLP-1 treatment. And actually, this anti-obese effect indicates one more nice area and interesting disease. It's called NASH. which is linked at the same time to inflammation, cell death, and excessive fat, fat liver disease, or non-alcoholic steatohepatosis. And we did experiment on mice in SNC company in Japan on this NASH model. And SKQ also worked nicely by several parameters. So if not obesity, then maybe fat liver disease, which is still popular, in brackets, indication for biotech drug development. So we hope that certain advantages of our drug, and mainly it is muscle protection and oral administration, will help our drug to compete or complete maybe GLP-1 agonist treatments. At the moment, as I said, we are fighting for more than almost 2 years trying to raise money for clinical trials, and we just closed the deal. So I hope that in half of the year, 9 months, we will start clinical trials in the United States at phase 1. But since it's obesity, I hope that some of our volunteers will be overweighted. So we'll get some first efficiency signals right from the first phase 1 of clinical trials, and we don't have conference next year, right? But in 2027, I hope very much to tell you from this stage of the result how this first mitochondria-targeted drug, does it work in humans or not. So that's more or less it, and thank you for your attention, and we have some time for questions.
When you're giving that, your product, what does it do to the, if you measure, to the anaerobic threshold or the VO2 max? Do it on mice and rats, you know?
It's difficult to measure on animals. Actually, it obviously it stimulates energy expenditure. So, and it shifts RER, so you can really see how they are burning fat and not carbohydrates, not glucose. But we don't see any effect on the oxygen level in the blood. But
not in the blood. Anaerobic threshold is...
and I see.
Anaerobic threshold and the VO2 max.
Actually, these are very robust parameters, so we don't change them much. You can measure it in, for example, isolated mitochondria, but it's unlikely relevant to the organism. So I don't have a solid answer to your question. Sorry.
More questions?
Okay.
No. Okay.
Thank you very much.
Thank you very much, Maxim.